So the RH factor, the first time it was identified in the rhesus monkey. So that's why what do you say it as a RH factor and the name of rhesus monkey. So that I already said that the people who have RH factor are said to be RH positive, and people who don't have RH factor, they can be called as RH negative, RH positive and RH negative. Okay. So then, in general, we show this particular RH positive, RH negative. We are seeing now. So in general, when we talk about the inheritance pattern, we use D. The word D. That means the letter D, capital D, indicates RH positive. Small d indicates RH negative. Okay, so let me write here. This is RH positive. This is RH negative. So when RH positive male or female, no matter, because it is autosomal inheritance. RH positive father, RH negative mother. So what you can expect here? So gametes I am taking. So what we can expect? The baby with RH positive or RH negative. So the baby is RH positive. That means we need to understand the dominant one. This is the dominant one. Capital D is the dominant one. So RH positive father. You can change even no matter. Positive mother married RH negative father. So this is mother and this is father. So even you get the same. That means there is no change here. This is a autosomal inheritance. So RH positive baby, son or daughter, whatever it may be, RH positive. So pattern of inheritance just correlate with the Mendel that this is tall plant and this is dwarf plant. What you obtain in the first generation, all plants are tall. So that's it. In that way we are talking about. Okay, that means it follows the law of dominance. RH factor is following law of dominance. So this is about the RH factor. What we said on about the RBC, a particular factor which is identified. Okay, so this particular factor is identified in the RBC. In some people it is present, in some people it is not present. That's all. In some people they don't have RH factor. So those who are having RH factor, we say them as RH positive. Okay. Then those who don't have RH factor, we said RH negative. Their genetic format can be shown like this: capital D, or capital D, small d. That means it is available in the dominant homozygous form and dominant heterozygous form. Okay, now here it is available only in the recessive form, absence of negative. That 90 to 95% of the people in the world are RH positive only, no doubt. That male or female, no matter. Then the remaining 5 to 10% of the people in the world are RH negative. That means definitely 90 to 95% is having this kind of factor. It doesn't make any energy, it doesn't give any energy, it doesn't improve the oxygen transport. Then why they are learning? Because a very interesting the pattern of inheritance is going to give the different kinds of result. What do you say that interesting result we are talking about is erythroblastosis foetalis, a kind of disorder. If you don't notice, it leads to erythroblastosis foetalis or hemolytic disorder of newborn baby, which is a very famous one that we are going to discuss now. Erythroblastosis foetalis or hemolytic disorder of the newborn baby. We can say it as erythroblastosis foetalis, or also it is called HDNB, hemolytic disorder of newborn baby. So how will it come? In which conditions it will come? That we have to discuss now. Hemolytic disorder of newborn baby. A disorder which is that some incompatible marriages. So what are the incompatible marriages that we have to see now? Okay. So let us see. Let us focus on erythroblastosis foetalis or hemolytic disorder of newborn baby. So in the case when father is positive, when father is Rh positive, when the mother is Rh negative, this is the case. We can say father should be positive. That means on his RBC, that particular factor is present, Rh positive. Mother is Rh negative. On her RBC, that factor is absent. We call it her as Rh negative. Okay. This is the incompatible marriage. Let us prove this. This is an incompatible marriage. So what happens? Let us see here. Let me show you. So in this case, capital D, father, small d, mother. For your understanding, I am even drawing that RH or RBC also here. That means the father is positive, which he contain a factor called RH factor, and mother is negative, which on her RBC there is no RH factor. So let me use another color for RH factor. So this is the condition. Okay, so this is father. Father. Why I'm showing again and again, because most times this is going to be the question here. In which condition erythroblastosis foetalis come? When the father is Rh positive, when the mother is Rh negative. So I am writing here that also. Rh positive. Father is Rh positive. Okay. So that RBC, there one I wrote. So mother is Rh negative. Okay. So father is Rh positive, mother is Rh negative. What we say about the baby? Positive or negative? Baby ఏంటండి పాజిటివా నెగటివా?
Positive.
Yeah,
Positive.
very good. Baby is going to be positive. Okay. So that means let us see what are the consequences here. That first day, no problem. Zygote, only chromosomes are present. But zygote is dividing 2 cell, 4 cell, 8 cell. So that leads to the blastula, marul, the gastrula. Likewise, when the baby is developing, that leads to the baby also form the blood vascular system. The baby also form their own circulator. That means 6th month, 7th month. As long as we are going, that the thing, what happens here? The baby is Rh positive. What type of RBC she produce? Rh positive type of Rh positive type. That means baby RBC contain Rh factor. I am saying here. Mother RBC doesn't have Rh factor, but baby RBC is having Rh factor. Now here we need a little knowledge of that mother placenta, knowledge of placenta, endometrium layers of the mother, and the blood capillaries of baby. So that means the blue and red I am showing here. That imagine the baby blood capillaries of blue and mother blood capillaries are red. That both can form the placenta. They arrange like an intricate net. So that the transmission of the oxygen and nutrients continuously supplied from the mother to the baby. So what happens during parturition period? That these blood capillaries from the mother, blood capillaries of the mother, red one, the blood capillaries of baby are going to be ruptured out. This is baby, I'm saying, ruptured out from the placenta. These blood capillaries are ruptured out, and these kind of RBC are enters into the mother. The baby RBC are entering into the mother. What is the speciality of baby RBC? They contain Rh antigen. Actually, mother don't know what is Rh antigen because she don't have. As we said from the beginning, if you don't have, you will consider it as antigen. That means for mother, this factor is new. That mother feels it as it's a bacteria, or it's a virus, or it's a protozoan, helminth, whatever foreign body. So she consider it as antigen. Okay, so when she consider this particular RBC, that means when during the parturition time, the placental capillary rupture makes over some of the RBC from the baby placenta enters into the mother. Okay, so that particular looking towards that particular factor, our regular protocol will happen that we learned in immunology. I'm giving a brief here also. T helper cells. recognize any foreign body which comes into our body. So that it means this is also foreign body. That's why they are recognized by T helper cell of the mother and the B lymphocytes. They inform to the B lymphocyte, and B lymphocyte will examine them, and they produce two types of cells. They differentiated into two types of cell. We call them as plasma cell, memory cell. Imagine this antigen for any parasite, the same mechanism. This antigen, if it is a lock, the plasma cell B lymphocyte should produce a key. Okay, the first time production of key is going to be comparatively late because they have to examine them. They need to make some similar kind of keys. We call the keys as antibodies. So that means simply mother considering that particular factor as a bacteria or virus, she started. Their immune system protocol begin. T helper cell recognize, inform to the B lymphocyte, and they start producing plasma, differentiating into plasma cell and memory cell. And plasma cell produce antibodies. What for what? These antibodies which are produced by the mother should go into the baby through the placenta, should destroy this kind of RBC. The sample came, then they produced antibodies to destroy the baby RBC. Okay, so for that they're running actually, but actually the production of antibodies for the first time is late. That's why the first child is escaped. Till mother produce antibodies, the first child will escape. Now the second child, the case, second child. Let us see. What about second child? Second child is also Rh positive. Father is positive, mother is negative. That's why second child also Rh positive. But you remember, for the first child, when she exposed, she produced memory cell, which rapidly they act now. Okay, the same case, zygote divides, blastula, gastrula, likewise, embryo is formed, a good enough placenta is formed, and again the same after the rupturing, some RBC of the second baby reaches to the mother, second baby also reaches to the mother, and that particular antigens, Rh antigens, mother feel them as antigen. They will be recognized, and now who act? Memory cell. First time, why? But first child is escaped because production of antibodies by the mother, the first time is late. Okay, so the second time you just forget about this particular father, because all these events happen in mother only. So then memory cells are present, they immediately produce the antibodies. That antibodies will come, enter into the baby, and they destroy, they start destruction of baby RBC. When they start the destruction of the baby RBC, that means bone marrow will try to produce large number of RBC, because mother antibodies are destructing the antibodies of the baby. So that's why baby will try to produce new antibodies. In the mean period of time, due to the rapid production, immature RBC will be produced over immature RBC. That immature RBC are called as erythroblasts. That's what we say, erythroblastosis foetalis. Immature RBC are produced over, because you're killing continuously baby RBC. The baby is trying to produce, but that no time we are giving. So that's why that immature RBC are getting produced. So what is the meaning of erythroblastosis here? Erythroblast. That means you need to understand they are immature RBC. They are immature RBC are produced in the baby. Finally, it leads to the death, no doubt. Next, that what I said, and one more word I given, hemolytic disorder. Hemo, RBC, lysis, destruction of whom? Newborn baby. Hemolytic disorder of newborn baby. Second child, when this incompatible marriage, the first child may escape due to the production of antibodies late for the first time, but second child, third child, fourth child, simply we say second child onwards, they die. So simply we define erythroblastosis foetalis like this. If the father is Rh positive, mother is Rh negative. If the father is positive, mother is negative, then from the second child onwards, the destruction of the RBC occurs and the baby dies, we call it as hemolytic disorder of newborn baby or erythroblastosis foetalis. This is about the erythroblastosis foetalis, the story of erythroblastosis foetalis, which we are seeing now. So that I'll give a brief here. I'll give a brief here with this, with clear visuals. The father is positive, mother is negative. That means mother RBC doesn't have RH factor. The baby is positive. So what happens? The baby is growing in the mother. When mother blood exposed to these baby RBC in the during the development, because gestation occurs in the mother only. So in the 9 months of time when it is happened. So these baby RBC, when through the placenta, when they reaches the mother, mother consider them as antigens like bacteria and virus. They treat, they produce the antibodies, and that antibodies, IgG antibodies, smaller antibodies, will they go into the mother blood and they destroy all the RBC here. But first production of antibodies for the first time is late. That's why the first child escape. First child is escape. But second child onwards, second, third onwards, they die because mother produce memory cells also. So that what do you say? It has erythroblastosis foetalis or hemolytic disorder of newborn baby. We say it has hemolytic disorder of newborn baby. Okay. So the very important concept here. From here you have to remember the second child onwards they die. Okay. So what kind of treatment will be given here? What kind of treatment? That means second child onwards we die. Okay, the technology, the science made us to know that this is an incompatible marriage, but they married. What to do? What to do? Now let us talk about what to do. What's the problem here? What's the problem? Let us try to see. So when particular RBC, okay, let me show you the clear diagram here. So that how do we solve this particular problem? Let us see here. How did we solve? How does the second child also saved? Okay, so the baby is RH factor containing baby. These RBC, when they enter into the mother, what we said, the T helper cell, and they activated the B lymphocyte, they differentiated into plasma cell and memory cell. They start producing antibodies. Memory cell will recognize this or that antigen. When they come for second time, also they destruct, isn't it? This is all the story we'll end over. Now, the first child, as I said, first child will escape. So after giving birth to the first child, to the mother, to the mother, that anti-D injection will be given over. After the parturition only, after giving birth to the first baby, to the mother, anti-D injection is given. What is present in the anti-D injection? RH antibodies are present. Anti-D injection contain RH antibody. What they do? That our injection given, that we are given the injection, our injection will work on baby RBC who are present in the mother. They destruct the RBC. Okay, mother is also doing. So why you have to give injection for the destruction of baby RBC who are present in the mother? Why you have to distract? Simple. If mother distracts, mother develop memory cells. It is going to be dangerous to the second baby. Then after that, whatever the baby RBC which are present in the mother, by giving anti-D injection, if you are distracting, you are not giving chance to the mother. So that means you are not giving chance to the activation of immune system in the mother. So that facilitates at least second child will be survived. That what about the third child? If she wants third child, same again. After second child, you have to give anti-D injection again. You have to give anti-D injection again. You that simple what we are doing here. Instead of mother destruction, we are doing destruction of baby RBC who are present in the mother. Don't give this injection to the baby. It is very dangerous, isn't it? They destruct the RBC, a first child even. So that's why. So just be careful here. That means we are going to get these kind of questions in examination. So you should think logically to answer all these. Okay, so this is erythroblastosis fetalis along with the treatment. Okay, then hemolytic disorder of newborn baby or erythroblastosis fetalis. We use it to say this is a condition. So the very very important thing you need to remember, this is happen due to a incompatible marriage, which is father is positive, mother is negative, second child onwards, that there is a possibility of disease which leads to the death also. What do you call the disease as? Erythroblastosis fetalis or hemolytic disorder of newborn baby. So this is the story. Okay.